Well, thank you. It's a pleasure to be here. I'm Russ Swerdlow. I'm going to discuss mitochondria and mitochondrial cascades in Alzheimer's disease. But first, what is Alzheimer's disease? It's been over a century since Alois Alzheimer made the association between progressive dementia and plaques and tangles. We've learned then that the plaques contain a protein, beta amyloid, that derives from a larger protein, the amyloid precursor protein, or APP. And APP has been implicated in Alzheimer's through genetic studies. We've learned that the tangles contain the tau protein, which phosphorylates and forms tangles. We know that variations in the ApoE gene correlate with lifetime Alzheimer's risk. Sounds like we know a lot about the disease, but do we? It really is a definition in flux. It is now, it always has been. It was originally defined as plaques and tangles and dementia in the young old. And then in the late 1970s, early 80s, there were a lot of work done here at UCSD. The definition was expanded to include plaques and tangles and dementia regardless of age, which made it incredibly common since age is the greatest risk factor. And then in the biomarker era, there's been a trend to try and diagnose the disease earlier and earlier on in the spectrum. For example, mild cognitive impairment, which is often a precursor to Alzheimer's disease clinically, in the presence of a positive biomarker, can be said to be Alzheimer's disease. And you can see that here in these Jack curves that show the evolution of biomarker changes as we currently measure them. And there's even been a proposal recently to diagnose Alzheimer's disease at this stage here, just in the presence of a positive A-beta biomarker. Now, I can see that it might be tempting to propose that, yeah, the plaques cause the tangles, which cause neurodegeneration and cognitive decline. But none of this tells us what drives the biomarkers. And I would propose that it can be a little dangerous to assign causality through correlation. And there are a lot of other issues that need to be addressed when we think about Alzheimer's disease. One of them is metabolism, energy metabolism, glucose utilization. That was shown to be a problem back in the early 1980s on fluorodeoxyglucose PET scans that showed glucose utilization in Alzheimer's brains was impaired. Now, when I first got involved in Alzheimer's research in the mid-1980s, it was in a lab that was studying the biochemical correlates of reduced brain glucose utilization as demonstrated on FDG PET. And the way we did that was to take autopsy brains and homogenize them and add the homogenates to a sealed vial and add radiolabeled glucose to the homogenate. The glycolytic enzymes in the homogenate would convert the glucose to pyruvate, and that radiolabeled pyruvate would access mitochondria and the Krebs cycle. Radioactive carbon dioxide would be released and captured in a bowl. And then we could transfer the bowl to a scintillation counter and determine the amount of radioactive CO2 that had been collected and determine rates of glucose utilization. We found that brains from young individuals consume the most amount of glucose. Brains from elderly, cognitively normal individuals consume somewhat less glucose, and brains from Alzheimer's patients still less glucose. We weren't sure why this was happening. At the time, we thought maybe it was due to a problem with glucose transport across the blood-brain barrier. It got me thinking, perhaps we can do an end run around this problem with glucose utilization by adding an alternative bioenergetic substrate, such as the ketone body beta-hydroxybutyrate. So I studied that for the next several years, except now when I used a similar approach, except now when instead of adding radiolabeled glucose to the homogenates, I was adding radiolabeled beta-hydroxybutyrate, which would access the mitochondria in the Krebs cycle and release radiolabeled CO2. And we found that Alzheimer's brains could utilize beta-hydroxybutyrate, although not at the same rate as control brains. But one thing that stood out is quite interesting, I thought, was that the rates of beta-hydroxybutyrate and glucose utilization in the Alzheimer's brains was fairly equivalent, which raised the question of an area of common pathology where their metabolisms overlap. And where their pathways overlap is in structures called the mitochondria. What are mitochondria? In high school, we learned that they are the powerhouse of the cell. They make energy. And energy is important. It's the spark of life. We need it to combat entropy. They can make energy because they've evolved a respiratory chain that harvests electron energy and uses that electron energy to make ATP. Mitochondria have their own DNA, the mitochondrial DNA, which does not play by the rules of Mendelian genetics. It's exclusively maternally inherited, and cells have many copies of mitochondrial DNA. All this copies of the mitochondrial DNA can be the same, that's called homoplasmy, or not, that's heteroplasmy. And cells can increase or decrease their mitochondrial DNA copy number. The mitochondrial DNA encodes 13 proteins, but there are over 1,000 proteins located at the mitochondria that form the mitochondrial proteome. Most of them are encoded on nuclear genes, and the proteins are imported to the mitochondria, and they're detailed in databases such as the mitocarta. And mitochondria are important in aging. They're one of the recognized hallmarks of aging. Mitochondria certainly are recognized as being altered in Alzheimer's disease. And if you look at the brains of Alzheimer's patients, the mitochondria simply look different. There's less mitochondrial turnover. There are reductions in various mitochondrial enzyme activities, and there are changes to the mitochondrial DNA as well. And we recognize in 2024 that beta amyloid localizes to mitochondria, and it may directly or even indirectly interfere with mitochondrial function to enact a host of downstream changes that I've written out here. This is what I refer to as a secondary mitochondrial cascade, and it's quite popular within the Alzheimer's field right now. I suspect because it exists within the confines of the amyloid cascade hypothesis that proposes that beta amyloid is responsible for everything that goes wrong in the disease. But there are some issues with this perspective, perhaps the main one being that changes to mitochondria in Alzheimer's patients are not brain limited. We see these problems in platelet, muscle, and fibroblast mitochondria, where they're harder to explain away as being a consequence of plaques or A-beta or tangles or tau protein. I think this really raises issues of cause versus effect. Mitochondria also relate to other phenomena that are recognized to occur in Alzheimer's disease. For example, insulin resistance. So muscle mitochondria are a recognized contributor to insulin resistance, and there is insulin resistance in the brains of Alzheimer's patients. Some have even referred to it as a type 3 diabetes. Neuroinflammation, of course, occurs in Alzheimer's, and mitochondria can trigger inflammation. For example, in microglia, there are two pathways, the NLRP3 and the cGAS-STING pathway, that are triggered by the presence of double-stranded DNA in the cytoplasm. And under sterile conditions, the most likely contributor of double-stranded DNA to cytoplasm is the mitochondrial DNA. Lipid biology is altered in Alzheimer's, and it's very hard to separate lipid biology from mitochondrial biology. The mitochondria launder the carbon that goes into fatty acid and cholesterol synthesis, and phospholipid synthesis plays out at the interface between mitochondrial and endoplasmic reticulum membranes in structures called the mitochondrial associated membranes. Now back to my story, I did my postdoc in the lab of Davis Parker. Doug, I know you knew him back in the day. And Davis had this very interesting hypothesis about sporadic neurodegenerative disease, in which he proposed that if you see what looks like a sporadic disease, but there seems to be a genetic component, consider maybe there's non-Mendelian genetics at play, such as mitochondrial genetics. And he had recently reported that the activity of a mitochondrial respiratory chain enzyme, cytochrome oxidase or COX, complex 4, that its activity was reduced in Alzheimer's patients. This is an interesting holoenzyme. It has 13 subunits. 10 of these subunits are encoded on nuclear genes, and 3 of them are encoded on mitochondrial DNA genes, which you can see here in red, CO1, CO2, and CO3. And we hypothesized that mitochondrial DNA would contribute to reduce cytochrome oxidase activity in Alzheimer's patients. And the way we tested this in the mid-1990s was to adapt what was then a recently described approach called the cytoplasmic hybrid or CYBRD technique, which I'll walk you through. You take a cell line, we'd like to work with neuronal cell lines, and here's a nucleus in blue, mitochondria with mitochondrial DNA in red. In blue are the mitochondrial, in blue are the nuclear encoded respiratory chain subunits, and in red are the mitochondrial DNA encoded subunits. Now, it's possible to completely eliminate the mitochondrial DNA from these cells. When you do that, of course, you eliminate the mitochondrial DNA-encoded respiratory chain subunits. You have a respiratory incompetence cell, and it's called a row 0 cell. Why is it called a row 0 cell? Row DNA is another name for mitochondrial DNA. So row 0 just means no mitochondrial DNA. And it's possible to keep these cells alive and in culture. We would then take a tube of blood from an individual. We would centrifuge out the platelets. Platelets are great for these experiments because they have mitochondria and mitochondrial DNA, but they lack nuclei. And we would add platelets and row 0 cells to a tube together. We would add a detergent to rough up the membranes and mix. And then we would quench the detergent. And we would end up with cells that had a mixing of the cytosols, so cytoplasmic hybrids or cybrids. And the mitochondrial DNA from the platelets would replenish the mitochondrial DNA within the cell and restore the respiratory chain. And so you can use these cell lines in biochemical molecular assays. So if you make a series of cybrid cell lines where the mitochondrial DNA comes from an Alzheimer's patient, and a series of cybrid cell lines where the mitochondrial DNA comes from control individuals, and if there are differences in biochemical or molecular function, these differences are reflective of differences in the mitochondrial DNA because the nuclear background is the same in all the cell lines, as is the environment that we grow the cell lines in. And when we did this experiment, we saw that we could indeed transfer reduced cytochrome oxidase activity to the cybrids with the transfer of mitochondrial DNA from the Alzheimer's patients, their platelets. We saw other changes as well. The cybrids containing mitochondrial DNA from Alzheimer's patients contain increased amounts of these large misshapen mitochondria with disrupted cristae. They perform more reactive oxygen species, so they had more oxidative stress. The mitochondrial DNA copy number did not quite come back up to the same level as it did in the control lines. After studying this model really for close to a decade, I began to think of things from the perspective of what I refer to as a primary mitochondrial cascade, which I'll walk you through. So the idea is that everyone inherits a state of baseline mitochondrial function that they inherit through nuclear genes that they get from their dads and their moms, and mitochondrial DNA that you get from your mom. So both parents contribute, but your mom contributes a little bit more. And it's fairly well accepted that as we age, our mitochondria become less efficient. And where this hypothesis really begins to deviate from the amyloid hypothesis is that this hypothesis is proposing these age-related mitochondrial changes trigger the proteostasis changes that we see in Alzheimer's, the plaques and the tangles, and the systems changes, some of which I mentioned, like insulin resistance and altered lipid biology and microglial activation. Now, these systems changes and proteostasis changes may feed forward to further impair mitochondrial function or not, but simply just with the passage of time, someone's mitochondria will continue to decline, and the person may reach a point that they can no longer compensate. And I would propose that when they transition from a state of compensated bioenergetic function to decompensated function, that's when they begin to experience the phenotype of Alzheimer's disease. Now, I recognize fairly quickly if I was going to present an idea like this to the Alzheimer's field, best link it to the classic histopathology changes that are associated with the disease. So I'm going to share some of those data with you. These are the first set of data I'm going to show you are from mitochondrial DNA depleted neuronal cell lines, the row 0 cell lines that I told you about earlier. Okay, by way of, in terms of background, I'll talk first about APP and beta amyloid. So APP, as I mentioned, has been implicated in Alzheimer's through genetic studies. Mutations in APP are associated with familial autosomal dominant Alzheimer's, presenilin mutations as well, and the presenilin gene products process the APP protein. But despite all this knowledge, APP's functions are still incompletely understood. A beta, of course, has been tied to Alzheimer's through the plaques. It's been proposed to cause Alzheimer's. That's the amyloid cascade hypothesis. It's been proposed that even without cognitive decline, someone can be diagnosed with Alzheimer's pathological change if they have plaques, or perhaps even now just diagnosed with Alzheimer's disease. It's the basis for an Alzheimer's biomarker. So in spinal fluid and plasma, levels go down, but A beta 42 levels, the 42 amino acid species, goes down more than the 40 amino acid species. And so you get this reduction in the A beta 42 to 40 ratio. And the idea is that neurons constitutively secrete A beta and it ends up in the spinal fluid. But when there are plaques present, the A beta gloms onto the plaques. And since A beta 42 is less soluble than A beta 40, more of it gets glommed onto the plaque, and you get this reduction in the ratio. This may be correct or not. I don't know. APP affects mitochondria. APP has a mitochondrial targeting sequence, and it's been reported that the protein localizes to mitochondria, begins to translocate into the mitochondria, and undergoes this translocation arrest. It can be processed to A beta at the mitochondria, and when APP is at the mitochondria seem to function differently. But it works both ways. In our mitochondrial DNA-depleted neuronal cells, we see this robust increase in APP mRNA expression and APP protein levels. And mitochondrial function seems to affect trafficking of APP in the neuronal cells and A beta release. What we found is that these neuronal cells produce APP, and when the mitochondria are functioning normally, most of the APP ends up at the plasma membrane, and A beta secretion rates are relatively high. But when the mitochondria are dysfunctional, like in the Rho zero cells, much of the APP siphons off to the mitochondria, less APP makes it to the plasma membrane, and A beta secretion rates go low. And we suspect that this may account, at least in part, for why A beta levels are reduced in Alzheimer's spinal fluid. And there are even some of us in Kansas who would propose that A beta may serve as a biomarker of mitochondrial function. What about the tau and the tangles? So the tau, of course, binds to microtubules, the cell cytoskeleton. There are different tau isoforms dependent on transcript splicing. When we're forming in utero, our tau is heavily phosphorylated, but as we, during normal aging, it's less phosphorylated, and then in Alzheimer's, it begins to phosphorylate again. It forms oligomers and fibrils, especially in its hyperphosphorylated form. And it's also used as an Alzheimer's biomarker. Extracellular levels rise. It's been proposed that this reflects tau release from dying cells, but it's certainly going to be more complicated than just that. Tau is also actively secreted by cells. Regardless of how it's happening, spinal fluid and plasma phosphorylated tau levels increase in Alzheimer's. Tau has been recently reported to co-localize to mitochondria, where it interacts with mitochondrial proteins to affect respiratory chain function. But it works both ways, of course. This is a Western blot from our mitochondrial DNA-depleted neuronal cells, and we've stained for tau. And you can see that the Rho zero cells have a lot more tau than the wild type cells. It's present here in a schmear. You can clean up the schmear by treating the lysates with a phosphatase prior to electrophoresis, and that really brings out the different splice isoform expression. We also saw in these cells an increase in oligomerized tau. And notice this band up here. We spent a lot of time thinking about this band. What does it represent? It's at about 100 kilodaltons, which corresponds in size to an isoform of tau called big tau that normally isn't expressed in the central, well, until recently wasn't suspected to be expressed in the central nervous system, although that's recently changed. And what we found was that if we look at tau expression in the mitochondrial DNA-depleted cells, there is an overall increase in tau expression. It's largely being driven by an increase in the expression of 4R-containing transcripts, including a 4R-containing transcript that also contains exon 4A and 6 expression. So this is a big tau transcript. And you can see that actually visually on the RNA-seq data, where you can see here that we're getting reads over exon 4A in the Rho zero cells, but not in the Rho plus cells. What about ApoE? So ApoE encodes a 299 amino acid peptide that forms lipoproteins with fatty acid and cholesterol. It's normally expressed in hepatocytes and astrocytes, although stressed neurons can also express it. There are 3 isoforms, ApoE 2, 3, and 4, that are defined by the amino acid that's present at 2 different spots, 112 and 158. And for reasons that are unclear, those who have an ApoE4 allele have a higher lifetime risk of Alzheimer's, and those who have an ApoE2 allele have a somewhat lower risk of developing Alzheimer's relative to the most common variant, which is ApoE3. ApoE can affect mitochondria, and this is work from Robert Maley's group. So he's reported that ApoE4 folds differently than ApoE2 and ApoE3. He's reported that in ApoE4, you get this salt bridge forming here, which pulls in this hinge, and this predisposes the protein to a proteolytic cleavage of its C-terminal 27 amino acids, which opens up a mitochondrial targeting sequence. And he's been reported that this derivative goes to the mitochondria and interferes with the respiratory chain. But it works both ways. If you look in our mitochondrial DNA-depleted neuronal cells, we see this robust upregulation of ApoE mRNA expression, and it carries forward into ApoE protein that we see being released into the medium. Remember, this is a primarily secreted protein, but it also begins to build up intraneuronally as well. Now, the fact that this primary state of mitochondrial dysfunction is altering the expression of genes in the nucleus raises this issue of mitochondria nucleus crosstalk, which basically means that what goes on in the mitochondria affects what goes on in the nucleus. And you can see this play out across a host of omics applications, such as RNA-seq, which we use to determine the entire transcriptome of a group of cells or tissue. And the way we analyze the data is by grouping genes into related pathways or groups or modules. This is the Alzheimer's domain from the KEG database being applied to RNA-seq data from our mitochondrial DNA-depleted neuronal cells. Where you see green, the expression of a gene or genes is going down, and where you see red, the expression of a gene or genes is going up. You can see here that there are a lot of genes in this Alzheimer's pathway whose expression levels are changing to the extent that we can say with statistical certainty that primary mitochondrial dysfunction alters the Alzheimer's pathway, at least as it's defined in the KEG database. But really what happens is that when you have this state of primary mitochondrial dysfunction, it reshuffles nuclear gene expression. So using a very liberal cutoff for what are differentially expressed genes or DEGs, we saw that in one row zero cell line, 84% of the genes showed differential expression. In another row zero cell line, it was 77%. We can apply a more stringent cutoff for what are significant genes, but it still remains very high. And if you do pathway analysis, there's just a tremendous number of pathways that are altered. Many of these pathways are pathways that are altered in Alzheimer's disease. I've been telling you about data from a model of mitochondrial DNA depletion. So I think I should say a few words about mitochondrial DNA copy number in Alzheimer's disease. Mitochondrial DNA, our group, as well as other groups, have shown that mitochondrial DNA copy number is reduced in the brains of Alzheimer's patients. I'm going to show you some of these data that were driven by my colleague, Shay Andrews at UCSF. Judy Poff here at UCSD was also a co-author on this work. But what we reported here, looking at brains from the AMP-AD consortium, that there is this inverse correlation between the degree of Alzheimer's neuropathologic change, plaques and tangles, and mitochondrial DNA copy number, such that the brains that had the highest mitochondrial DNA copy number had the least amount of Alzheimer's neuropathologic change. Conversely, we saw this direct relationship between cognitive performance and mitochondrial DNA copy number, such that those brains that had the highest mitochondrial DNA copy number those individuals prior to death had the best performance on their cognitive testing. You can also, from these brains, ascertain heteroplasmic mutations. That's what this circus plot is showing. Each circle on the circus plot is showing heteroplasmic mutations in different brain regions from different cohorts contributing to the AMP-AD consortium. You can see here that there is this massive accumulation of heteroplasmic mutations here in a region called the control region. And the number of these heteroplasmic mutations in the control region is in excess in the brains of the Alzheimer's patients. This is important because the control region has two main functions. It's important for mitochondrial DNA replication. So if there are problems with the control region, you may not be able to replicate your mitochondrial DNA. And it's important in mitochondrial DNA transcription, because that's where it initiates from. So I've been throwing around terms like mitochondria, aging, and compensation. Can I tie them together? Well, these are data that we recently reported in which we looked at the status of mitochondria in the brains of aging mice. What I'm showing you here is the mitochondrial DNA copy number in the hippocampi of young and old mice, and you can see that there is this increase in the mitochondrial DNA copy number in aging mice, and this is what happens in the brains of humans as well as they age. If you look at respiratory chain function, we actually did not see a clear decline, certainly not a statistically significant decline in respiratory chain function here for complex 1 and cytochrome oxidase, complex 4. That's when we're referencing the Vmax's, the activities of the enzymes, to the amount of protein in the assay. But if we then go and we reference those activities to the amount of mitochondrial DNA in the tissue, what we see is that there is this profound difference in the activities. What this indicates is that the mitochondria in the brains of the old mice have to work much harder to maintain the desired level of bioenergetic function. I try to capture this in this cartoon. So on this x-axis, I'm showing age, going from young to old. On this y-axis here, I'm showing function in terms of bioenergetics. And on this axis, y-axis here, compensation measured as mitochondrial DNA copy number. The gist is that the brain will maintain the level of bioenergetic function that it needs. But how hard does it have to work to do that? If you're using mitochondrial DNA copy number as your biomarker of how hard you're working, when you're young, not very hard. But as you age, it takes more work and you need to increase your mitochondrial DNA copy number. What happens if you can't raise your mitochondrial DNA copy number to levels needed to maintain biologic function? Well, then you lose function. You're decompensated now. And I would propose that this is the point where the wheels start to come off the wagon and people start to get into trouble clinically. Now, if you had asked me a few years ago, what's the difference between this group here and this group here, I probably would have said that, yeah, this group here loses its ability to maintain its mitochondrial DNA copy number. Now I see it a little bit differently. I suspect the issue is that this group here reaches its maximum level of compensation and experience decompensation. Whereas this group here managed to stay out of trouble because they can keep pushing up their mitochondrial DNA copy number to the levels that are needed to maintain function. Okay, so this is all kind of neat, I suppose, and a big criticism is that, well, you're studying an immortal cell line where the cells divide. So can you look at this in a better model? So we've been working on that. I'm going to show you some data from one model that we've been working on developing, generated from induced pluripotent stem cells, where we convert them into neurons, and then we induce mitochondrial DNA transcript translation. So we block mitochondrial ribosomes with an antibiotic, chloramphenicol. And when we do that, we can show that we're blocking the translation of mitochondrial DNA encoded proteins. So you can see these are two proteins that are encoded by mitochondrial DNA. Their protein levels are both down. This induces a compensation that we see in aging, which is the mitochondrial DNA copy number is going up. So it looks like you're trying to make more mitochondrial DNA perhaps to get over this block. And consistent with that is if you look at the expression levels of the mitochondrial DNA expressed genes, you see the RNA expression levels is going up across the board. Now, what is the story of APP and A beta in this model? Well, APP mRNA levels go up, APP protein goes up, the cells begin to secrete more A beta. Now, I know earlier we talked about in Alzheimer's, A beta secretion levels are actually going lower in Alzheimer's patients in their spinal fluid. But what's really interesting is that in the model, as we were characterizing it, in which the neurons were in chloramphenicol for 1 week, respiration did not go down. So the neurons were still in a state of compensation. Now, and so I suspect here that what we're what we're mimicking here is more brain aging in Alzheimer's disease. And so we've tweaked the model, and with just letting the cells sit longer in chloramphenicol, then we start to see the respiration come down. And I think that's probably going to be a better model of Alzheimer's. But what happens to tau in these chloramphenicol treated neurons? Well, MAPT mRNA levels go up, total tau goes up, and phosphorylated tau levels go up. Now, the phosphorylated tau levels may be going up just because total tau is up, but if you look at how the kinases are set that phosphorylate the tau, such as GSK3 beta, serine phosphorylation at serine 9 activates it to phosphorylate tau. We see relative activation of GSK3 beta in those cells. We looked at the impact on ApoE, and as we see in the mitochondrial DNA depleted neuronal cells, ApoE mRNA levels go up in these neurons. We see this increase in ApoE secretion, and ApoE protein builds up in the neurons themselves. We see both the full-length ApoE building up as well as this 12.5 kilodalton cleavage product of ApoE, and both of these are building up in the neurons. We see this interesting inverse relationship between the amount of ApoE within neurons and the mitochondrial mass in the neurons. So green is staining for ApoE, red is staining for mitochondria, and we see this inverse relationship where cells that have very little mitochondria have a lot of ApoE and vice versa. And just to throw in there, we believe that we're seeing a general problem with proteostasis. Alpha-synuclein mRNA levels go up, and alpha-synuclein protein begins to accumulate within the neurons as well. Okay. So in the last few minutes, I'm going to transition to talk a little bit about therapeutic implications. So our group is very interested in manipulating energy metabolism in mitochondria for therapeutic purposes in Alzheimer's disease. There are a lot of different ways to do this. One way that we've been interested in is the ketogenic diet, which I talked about earlier. These are data that we published a few years ago in which we placed mice, just regular garden variety mice, on a ketogenic diet for 3 months, harvested their brains, isolated their neurons, and performed RNA-seq on the neurons. And we asked the question, what diseases would be predicted to be most likely affected by a ketogenic diet as when analyzed agnostically using RNA-seq? And interestingly, the disease that rose to the top of the list was Alzheimer's disease. The reason being that in Alzheimer's disease, we see reductions in the expression of genes that support the insulin signaling pathway and oxidative phosphorylation. But in the neurons in the mice on the ketogenic diet, we saw this upregulation of the expression of genes that support oxidative phosphorylation and the ketogenic diet. And for that reason, Alzheimer's disease was identified as a disease that one would expect to be affected by the diet. We actually performed a pilot study of the ketogenic diet in Alzheimer's patients. We placed 10 individuals on a ketogenic diet for 3 months. These are people with Alzheimer's, and we ascertained their cognition using the Alzheimer's Disease Assessment Scale, or ADAS-Cog. In the ADAS-Cog, scores going in this direction are improving, and going down in this direction are declining. You can see here that there was one participant who was protocol non-compliant, who stopped for unclear reasons his cholinesterase inhibitor and memantine during the study. He lost ground during the study. There's one participant who neither gained nor lost ground. And the other 8 all showed varying degrees of improvement, some what looked like a rather robust level of improvement. And we frequently saw a pattern that looked like this, in which the participant would provide a baseline score, and then after 3 months on the diet, their score would improve, and then after a 1-month washout period, the score would revert back to baseline. And because of this reversion back to baseline, we suspect that this here may not simply represent a practice effect. And we're currently engaged in a much larger trial of the ketogenic diet, which by this time next year should be completed. So this is meant to be a summary slide. I call it a mitocentric view of Alzheimer's disease. The general idea is this: we get older and our mitochondria become less efficient, and we adapt to that. And as we adapt, part of the adaptation involves a retrograde signaling back to the nucleus that reshuffles nuclear gene expression. We get changes in the expression of genes such as ApoE. For example, ApoE expression may turn on in neurons, and that ApoE in the neurons, either as full-length ApoE or as a cleavage product, may feed back to the mitochondria and further interfere with mitochondrial function. And this may be more profound in those who have an ApoE4. So I hope I haven't overwhelmed you. Obviously, this is a topic that I'm quite enthusiastic about. I even thought about, at one point, getting a vanity license plate such as this, which I did not do at the last minute upon remembering how much I was ridiculed for being a nerd back in high school and college and middle school. But I'll leave you with this one thought. Even if these Jack curves inform your conceptual approach to Alzheimer's disease, consider that it may be playing out against a mitochondrial background, such that those who have less robust mitochondrial function may transition through these stages at a younger age than those who have more robust mitochondrial function. So I will stop there, and I think there's plenty of time for questions.